Hello, I'm Tim Sandle, and this is a presentation about PMAT Module 1, which is part of the Pharmaceutical Advanced Microbiology training. I am sorry I can't be with you today, but I've had some complications at my workplace, and that's keeping me away for today. However, I will be with you tomorrow for one-to-one workshops. So what I'd like to do in this presentation is introduce myself, then I shall run through the general PMAT format, then I shall have a look at the module layout, then we'll have a look at some of the key concepts and explore these with group exercises. Then I'll touch on the assignment, exam and some aspects of communication. So, first of all, my name is Tim Sandle and my main job is Head of Microbiology at Bioproducts Laboratory. I'm also a tutor with the PMAT program, and I tutor on the PMAT program modules 1, 3 and 4. I've been involved with the PMAT program since 2006. I also serve on one of the International Clean Room Committees, and I'm a long-standing member of PharmIG committee. PharmIG is the Pharmaceutical Microbiology Interest Group, which is the leading UK and Irish society for pharmaceutical microbiology, and I also do some journalism in my spare time. In general, the PMAT units, and PMAT 1 in particular, is designed to be about 150 hours of study, and that's done over a 15-week period. There's reading in the text, quite substantial chunks to read, and there are a series of activities and exercises. It ends with an assignment, which is a written essay, and then there is an exam. And I'll say something about the exam at the end of this presentation. Okay, just about some resources. So although there is core text provided, there is a website that you may find of interest, which is the Pharmaceutical Microbiology website, which has daily updates of hot topics in pharmaceutical microbiology. And there are also 2 books that are recommended to go with the course. One is Microbiology and Stability Assurance in Pharmaceuticals and Medical Devices, and the other one is a glossary of pharmaceutical microbiology terms. This is a hardback book. This one's only available as an e-book. I'll just let you have a look at those for a moment. Okay, so this module is designed to be the introductory module to the Pharmaceutical Microbiology program. And it's a mixture of theoretical concepts reinforced with practical examples. It's designed to bring you up to speed on microbiology, introduce the concept of pharmaceutical microbiology, and to outline the way in which the pharmaceutical industry works, everything from documentation to formulation to regulations, GMP and so forth. Now what I'll do now is just run through the core units, and I'll step away from the camera and go through the screens. After I've done that, we'll look at some of the key concepts, do some exercises, and I'll come back on screen when it comes in time for the examination. So, Module 1, Unit 1. This is an introduction to pharmaceutical microbiology. And this is a fairly newly written addition to this unit, and it will be used for students taking up this module from this September. Okay, so this is an introduction to the essentials of pharmaceutical microbiology. So it begins by discussing how pharmaceutical microbiology differs from other branches of microbiology, covers the main tests, and explains the primary concerns that pharmaceutical microbiologists face in their day-to-day activities. Okay, Unit 2 is an introduction to the pharmaceutical industry, and it's a broad introduction describing the key factors which have shaped the industry. The unit discusses how the pharmaceutical industry is structured and looks in depth about processes and constraints and the way that new drugs are developed and end up coming onto the marketplace. Unit 3 is about risk assessment and contamination control. So it looks about how risk assessment and risk management concepts can be applied to microbiology. It looks at methods of contamination transfer and explores contamination risk models. And this is designed to fit into out of limits investigations and risks to processes and so on. Unit 4 looks at raw materials and water. This looks at microbiology contamination to different types of raw materials, such as synthetic, natural, animal and so on. Looks at water activity, looks at the types of microorganisms that are found in water systems, and explains the different types of pharmaceutical grade water, such as purified water and water for injection, and how they are developed and created and should be monitored for. Unit 5 looks at the challenges, the microbial challenges to the pharmaceutical industry. So this is a basic primer about microbiology, looking at the different sizes of microorganisms, the metabolism, growth requirements, where bacteria, fungi are found. The unit addresses the way that microorganisms challenge the pharmaceutical processes and products, and particularly how the behaviour of people who work on pharmaceutical processes can affect the microbial burden found in the areas. The unit also looks at contamination spoilage in different pharmaceutical dosage forms, and then has a section on sterile products manufacture. Unit 6 considers environmental control, so here is an introduction to clean rooms, isolators and other forms of barrier technology, clean room classification. There's an introduction to environmental monitoring, and there's a look at aseptic techniques and clean room disciplines. Unit 7 considers disinfection and preservation. So there's an introduction to disinfection and why it's necessary. In here we also look at the different types of disinfectants, ways to assess the microbiological performance of disinfectants. We look at disinfectant efficacy testing, the principles of preservation and preservative efficacy testing. Unit 8 looks at basic microbiological laboratory techniques. So it focuses on the tests that are described in the pharmacopoeia, such as the microbiological examination of non-sterile products, tests for specified microorganisms, sterility testing, pyrogen and endotoxin testing, assays on antibiotics, and antimicrobial preservative efficacy testing. And these can be regarded as the core compendia subjects. Unit 9 considers aspects of manufacturing and validation. So it looks at manufacturing procedures, specification records, pharmaceutical process validation, and then draws all that together to look at the role of the microbiologist in validation and areas such as cleaning validation. The final unit of the module is GMP, Documentation and Regulatory Affairs. There's an introduction to GMP. There's aspects of documentation such as electronic records and how that fits into EU GMP and ISO 9001 requirements. And there's a look at regulatory affairs and regulatory controls, such as systems of drug registration, the role that the pharmacopoeias play in this process, and finally the regulatory aspects that relate to marketing a pharmaceutical drug product. Okay, so what is pharmaceutical microbiology? Well, it's a branch of microbiology. In many ways, some see it as an applied branch of industrial microbiology. and it's the microbiology discipline of relevance to healthcare and to pharmaceutical manufacturing. Okay, so it's time for an exercise. What I'd like you to do is to get into groups, ideally groups of about 2 or 3 people, and I'd like you to consider the question of what are the objectives of pharmaceutical microbiology? What are the core essences? What are the kind of things that a pharmaceutical microbiologist gets involved in? If you can spend 10 minutes on this exercise, when you've completed it, if you can have a classroom discussion, so one person from each group can feed back, and when that's done, I'll come back and share my thoughts and ideas. So if the person responsible for playing this video can now pause it, and you can go and do the exercise, and I'll be back with you shortly. Welcome back. So I hope that was an interesting exercise, and you found out some interesting facts about the aspects of pharmaceutical microbiology that you consider to be of importance. What I'd like to do now is share some of my thoughts with you. Okay, so these are some of the objectives that I've come up with. Okay, so pharmaceutical microbiology is about the study of microorganisms associated with the manufacture of pharmaceuticals. It's about designing ways to minimise the numbers of microorganisms found within a process environment. It's about ensuring that the finished product is either sterile, or if it's a non-sterile product, then it doesn't contain those microorganisms that we might regard as objectionable and could cause patient or customer harm. It's also concerned with toxins, particularly microbial byproducts. It's also about having sound sampling and testing regimes to assess the microbial quality of the pharmaceutical manufacturing process. So this involves the testing and sampling of starting materials, in-process samples or intermediate product, the examination of the final product formulation, and the testing of the final product. So with the starting materials and in-process samples, there's a number of test methods that we can draw upon. So there are tests for microbial enumeration, which we can divide into the total microbial count and the total yeast and mould count. There is also a presence-absence test for specific indicator organisms. So many years ago, these used to be called objectionables. Now they are microorganisms which may indicate a concern with an incoming material, and this then extends out to a whole range of organisms that could be objectionable to the process. And there is also then bacterial endotoxin testing, which we invariably conduct through the limulus amoebocyte lysate LAL methodologies. Okay, with finished products, well, we have final product bioburden if it's a non-sterile product, or the test for sterility if it's a sterile product. There is also, if the product contains a preservative, we need to do preservative efficacy testing, or if it's an antibiotic, there'll be antibiotic effectiveness testing. We also then may be interested in looking for pyrogens. This is primarily endotoxin. And we also have a concern for looking for visible particulates, which are often non-microbial, but often falls within the remit of the microbiology laboratory to ensure that the product is of suitable colour, appearance, stability and particulate free. Okay, also falling within the remit of pharmaceutical microbiology is an assessment of the environment, and this is clean rooms and clean room air systems. So it's maybe the wall-mounted HEPA filters or the localised unidirectional airflow devices, barrier systems, isolators and so on. Pharmaceutical microbiology is also concerned with the assessment of water systems. Okay, so pharmaceutical microbiology is about ensuring that healthcare products and pharmaceutical products are safe. And these are generally lumped under the term of medicine. A medicine, in this sense, is any substance or component that is administered for the purpose of diagnosis, treatment, cure, mitigation, or the prevention of a disease. The terms medicinal substance, drug substance and active pharmaceutical ingredient are often used interchangeably, and in the context of this introductory module can generally be regarded as the same thing. In all cases you're trying to make them as safe as possible from microbial contamination. Okay, with pharmaceutical microbiology, the essential tests are described in the three main international pharmacopoeia. These are the United States, European and Japanese pharmacopoeia, and they contain a number of chapters or monographs that describe some of the essential tests related to pharmaceutical microbiology. And these are the sterility tests, the bacterial endotoxin test, microbial enumeration methods, antimicrobial susceptibility testing, methods and limits for the testing of pharmaceutical grade water, disinfectant efficacy testing, pathogen and abnormal toxicity tests, environmental monitoring and biological indicators. There are other activities that pharmaceutical microbiologists are involved in which may not have direct pharmacopoeia chapters. Now, in the case of the United States pharmacopoeia, there are far more monographs in there than there are in the equivalent European and Japanese versions. But other activities that pharmaceutical microbiology is involved directly or indirectly include microbial identifications, tests for water activity, because water activity determines the possibility of microbial survival based on the relative dryness of the material being examined. Disinfectant efficacy testing, antimicrobial susceptibility testing, microbial immersion studies often linked to demonstrating whether a container closure is suitably intact to prevent microbial ingress, cleaning validation studies, the maintenance of microbial cultures. Microbiologists also advise on process and equipment design. They will conduct risk assessments of the manufacturing process. They will investigate outer limits results. They will also be involved in the maintenance of laboratory equipment and also have a role in laboratory training. So you can see the full varied nature of the role of the pharmaceutical microbiologist and how important and valuable it is for most pharmaceutical and healthcare companies. Okay, I'd like to move on to have a look at microorganisms. So microorganisms refers to the collection of organisms that share the single characteristic of being visible only with a microscope, so hence microorganism. This includes bacteria, cyanobacteria, fungi, unicellular algae, protozoa, viruses and so on. But generally, pharmaceutical microbiology is referring to bacteria and the fungi. Okay, so you see on the screen, and this slide may be a little bit unclear, but this is reproduced in your course notes. There are some diagrams, and I should just explain to you what they are. So we have shapes of microorganisms. So we have bacillus, long rod, coccus, or coccoidal, spherical shape. We have a spiral bacterium. We have a spirochete, the corkscrew-shaped bacterium. Vibrio, the boomerang-shaped bacterium. Then representative growth forms, we have a chain of cocci linked together as staphylococci may appear under the microscope. Then we have a clustering effect of cocci as micrococcus might appear under the microscope. Then a pair of cocci, a diploid, and then a chain of bacilli together. All these different forms that the microorganism can take, particularly when grown on microbial culture medium. And then we have the diagram of fungus. So you can see there interesting shapes, varied shapes. Okay, bacterial growth. So all bacteria grow in the same way. Growth begins with a lag phase, so in the first few minutes of bacteria being within a growth medium, there is no real change to cell number or cell density. Then there is a rapid onset of growth, and that's what's called the exponential or logarithmic phase. And here there are back-to-back division cycles, so it means that you have one cell becomes 2 cells, become 4 cells, become 8 cells, become 16 cells, 32, 64, and so on in relatively quick succession. And when you consider a bacterium like Escherichia coli, that cellular division is taking place every 20 minutes. So within a few hours you've got a very, very large biomass of cells. Then we have the stationary phase, which is where the rate of cell division equals the rate of cell death. This often happens when the microbial culture has exhausted all of the immediate nutrients. After that, we have the death phase, or logarithmic decline, which is when the bacteria are no longer multiplying and the cells are reaching their natural stage and then dying off through the lack of nutrients. And with that, you get the classic bell-shaped curve. Okay, so different types of microorganisms have different growth requirements that affect the way that classic curve is constructed. And these are growth factors and energy sources, which could be ions or particular vitamins. Then their growth is affected by temperature. A bacterium that likes to grow in the cold will stop growing when it's moved into room temperature, for example. pH, osmolarity, and that often relates to the salt content within an environment, and oxygen, whether the bacterium is aerobic, anaerobic, or within anaerobes there's obligate anaerobes and facultative anaerobes and so forth. Okay, but generally we can divide bacteria into 3 groups based on their temperature growth requirements. So we have a group that we call the mesophiles. And mesophile means moderate temperature loving. And this is where we have an optimal growth range of temperature between 25 degrees Celsius and 40 degrees Celsius. And these are probably the microorganisms at the greatest risk to pharmaceutical processing, because they're the ones that can grow readily inside the human body. Remember, the human body has a typical temperature of just under 37 degrees Celsius, so these bacteria will grow quite happily. Then we have the cyclophiles that prefer cold temperatures. Many of these have an optimal growth temperature of about 15 degrees Celsius, but some can grow as low as 0. Then we have the thermophiles, and these thrive in very hot environments and have optimal growth of around 50 to 60 degrees Celsius. And you may find these kind of organisms in, say, a compost heap. There are also extreme thermophiles or extremophiles that can grow at temperatures above 90 degrees, but you're only going to find these inside a geyser in Yosemite Park or in thermal vents deep in the ocean. Okay, let's turn our attention now to culture media. And it's time for another exercise.